Good morning, everyone. I'm Alice Defarge. I'm the Interim Associate Dean of Research and Graduate Studies at OVC. I'm welcoming you today for the Chapel Lecture Series. Thank you for being here. We acknowledge that the University of Guelph resides on the ancestral lands of the Ottawa-Darwin people and the treaty lands of the territory of the Mississauga of the Credits. We recognize the significance of the Dish with One Spoon Covenant to this land and offer our respect to our Anishinaabe, Haudenosaunee, and Métis neighbors as we strive to strengthen our relationship with them. Today, this gathering place is home to many First Nation, Métis, and Inuit people, and acknowledging them reminds us of our important connection to this land where we learn and we work. So this Chapel Memorial Lecture Series is held annually since 1988 and has hosted numerous internationally known scientists who have inspired and motivated our graduate students, our faculty, our staff to achieve the highest possible goals in basic or applied research and to inspire our graduate and undergrad students to consider alternate career and become leaders in their profession. So this lecture series was established in 1987 through an endowment from the OVC graduate Dr. Clifford Chappell and his wife in memories of his father, Herbert James Chappell. So I need some notes because you have an extensive CV. So I'm pleased to introduce our speaker for this year, Dr. Darwyn Kobasa, head of the High Containment Respiratory Viruses Group in the Special Pathogens Program at the National Microbiology Lab in Winnipeg, and adjunct professor at the University of Manitoba Max Rajji College of Medicine. He earned his PhD in biochemistry from the University of Alberta, then did a postdoc fellowship at St. Jude's Children's Research Hospital in Memphis, Tennessee. Then following this, he was at the University of Wisconsin for 3 years as a research associate. Then before taking this current position at the Public Health Agency in Canada in 2003. Dr. Kobasa leads a group focused on understanding viral pathogenesis and developing medical countermeasures for emerging zoonotic respiratory viruses, including influenza viruses, NEPA viruses, coronaviruses, and filoviruses. Sorry for my accent, I'm French. I can play my French card today. Dr. Kobasa's research program makes extensive use of animal models to study viral transmission and disease mechanisms, and to conduct preclinical evaluation of vaccines, antiviral drugs, and antibody-based therapies in the Containment Level 4 lab. Following the emergence of clade H5N1 avian influenza viruses in Canada, his group has worked in collaboration with the National Center for Foreign Animal Disease to characterize the virulence and transmissibility of novel viruses genotypes, and to evaluate innovative vaccine strategies aimed at improving preparedness for emerging viral threats. So I will give you the floor, Dr. Kobasa. We're very honored to have you at OVC today.
Thank you. Yeah, thank you for the introduction. And, it's really a privilege and an honor to be here today to give this lecture. So I really want to thank the organizers for the chance to come here and for also the chance to meet with our longtime collaborators last night. It was very nice. So I thought I would start out and just maybe reiterate a couple of the things that Dr. Defarge mentioned this morning about what we do at the NML. So just to give you an idea, we differ a little bit in our focus from academic research. So, I work at the National Microbiology Lab in the Special Pathogens Program, and we have a specific mandate that's directed towards the protection of the health of Canadians. We kind of first and foremost provide diagnostic services and surveillance for risk group 4 pathogens in Canada, and also extend that to a bunch of risk group 2 and 3 pathogens that are kind of orphaned, that other programs don't work with. We undertake research on risk group 3 and 4 pathogens to understand how the organisms cause disease. And most of this work is things that we do in animal models of infection, so we're well set up for that. We undertake research to develop strategies to combat these infections, so medical countermeasures, mostly like vaccines, antivirals and antibody therapeutics. We provide assistance, training, and support internationally during outbreaks. This has primarily been for Ebola virus over the years, so that's usually what we respond to when there's an outbreak. And then we provide training at the postgraduate, postdoctoral level as well. The Special Pathogens Program works out of the Containment Level 4 lab at the National Microbiology Lab. So we do all of our work with level 3 or 4 pathogens in this lab. And we have a very large virus repository of risk group 4 and 3 and 4 viruses that we use in our work. The program itself is divided into 5 units that are headed by each by a different PI and their group. And I work in the high containment respiratory viruses group. And because we work with emerging zoonotic pathogens, we interact very closely with the National Center for Foreign Animal Diseases, which is in the same building as we are. And that makes it very easy for us to collaborate because we're just kind of across the hall from one another. And for the last 3 years, I guess, I've been working very closely with Johannes Verheyen in the Avian Diseases Unit on the H5 outbreak. So the virus that we're looking at is this clade 2.3.4b H5N1 that originated in Europe and Asia, and in late 2021 was transmitted into Canada through the Atlantic flyway. And it was first isolated from backyard poultry and a Cooper's hawk in Newfoundland, and then it spread very rapidly across North America and all the way down into South America and eventually into Antarctica. In the first year of the outbreak in the US, there was around 58 million poultry deaths that are associated with the outbreak, extensive wild bird deaths, and mammalian infections, so infections in a variety of mammals. And then it spread across Canada similarly. There was a similar situation with 7.2 million poultry deaths and infection in a large number of wild bird and wild mammal populations that are not known to be normally susceptible to infection by H5N1 virus. So the host range of these clade 2.3.4b viruses is very wide, much wider than historically what was associated with H5N1 viruses. All type A influenza viruses that the H5N1 viruses belong to are natural pathogens of aquatic waterfowl. And they spread from these birds into other highly susceptible birds like poultry or other seabirds, where they're maintained by replication in that host. And it can also cause very severe disease in these animals. They are occasionally transmitted into mammals. In this case, with this virus, it was spread into a very large number of mammals. For the most part, mammals are dead hosts for the virus because they lack the receptor types and other characteristics that are required for transmission of the virus within the hosts. But this virus was notable because there were a few outbreaks in mammalian species where there was sustained replication within that species. The mink in Europe is one example, sea lions in South America, and most notably this outbreak that occurred in cattle in the United States. And from the cattle, there was spillover into a lot of other animals. There was about 70 associated human cases. And most of these cases are asymptomatic or mild diseases or mild cases of infection, although there were a few severe cases and deaths associated with the spillover into people. So I just want to give you a little bit of information about the virus, just to kind of give you some perspective for what I'm going to be talking about. It's a lipid envelope virus particle. It has 2 major surface glycoproteins, the hemagglutinin, or H.A, which is the receptor binding protein, so it attaches viruses to cells. It attaches to sialic acid, and the type of sialic acid is particular to the species of animal that the viruses bind to. There's 18 known subtypes, so H5 is just one of the subtypes that's known, and H.A is the primary determinant of the protective antibody response to influenza. So it's one of the prominent antigens that's always included in vaccines. Neuraminidase is the other major surface glycoprotein, and it's an enzyme that removes sialic acid. there is 11 subtypes, and NA is also a target of the antibody response, but antibodies to NA are not specifically neutralizing, but they will interfere with viral replication. And then a minor component on the surface is the M2 protein, which is an ion channel. And this has been a target in some ideas for universal influenza vaccines because it's very highly conserved between all influenza viruses. So that will show up in some of the vaccines that I'm going to talk about a little bit later. One of the other features that's very important about the influenza is that it has a segmented genome. So it has 8 individual gene segments. And this is very important because it allows the exchange of gene segments in different viruses in a process that's just termed reassortment. And finally, the other protein I'll mention is one of the polymerase proteins, PB2. And there are a number of other structural proteins, nucleoprotein, matrix protein, and some non-structural proteins that I'm not going to talk about. So when we're doing the work that we're looking at different virus isolates in animal models, and the purpose for doing this is to understand what changes may be occurring in the viruses that's going to increase the risk to the human population. So some of the things that we're looking for when we look at virulence in animals and associated genotype is reassortment patterns with other viruses. So these viruses, when they were transmitted into North America, quite freely reassorted with a number of avian viruses in the environment. There are other determinants of adaptation to mammals that are known. Receptor binding changes are thought to be the most critical change that would be required for the adaptation of an avian virus into a human virus. So we're always, of course, on the lookout for that. And then other mutations that enhance the fitness of replication of an avian virus for replication in mammalian cells. I'll occasionally mention this PB2E627K or the mutation that is important for the adaptation of avian viruses to human cells. So initially when we started working with Johannes, he had been sequencing virus isolates that were obtained from wild animal deaths. And this was a mix of birds and mammalian species. And they gave us some that they thought would represent the genotypes that they were observing in the wild. So we started with these 4 viruses, one from a blue-winged teal, one from a red-tailed hawk, one from a red fox that was isolated in PEI, and a red fox that was isolated in Ontario. They all have different gene kind of mixtures or constellations. The red fox PEI is all Eurasian, although it has this lysine mutation of PB2627, which is this characteristic mammalian determinant. The red-tailed hawk and red fox Ontario viruses also have this change in PB2, while the blue-winged teal virus has the avian signature at this position. So the animal models that we use when we're looking at characterizing viruses and how they may represent what's going on in humans are typically the mouse and the ferret. And the reason we use these is because of the different types of receptor binding that these human and avian viruses exhibit. So human viruses preferentially recognize alpha-2,6-sialic acid. Avian viruses preferentially bind to alpha-2,3-link sialic acid. So that's the key difference between avian and human viruses for receptor binding properties. With mice, they're only susceptible to experimental infection because they do have the avian virus receptors in their upper and lower respiratory tract. And they're used because differential virulence with H5N1 viruses typically matches virulence in mammals. But I'm not going to talk about the mouse data that we have today. I'm going to mostly focus on the ferret information. And ferrets are interesting because they're naturally susceptible to infection with human viruses. And this is because both humans and ferrets have an abundance of this human type sialic acid in their upper respiratory tract. They both have some mammalian receptors in their lower respiratory tract, so that is why humans can be susceptible to infection with H5N1 viruses, and ferrets can also be experimentally infected with H5N1 viruses or avian viruses. They have a similar course of clinical illness, and the transmission in ferrets is considered predictive of human transmission potential. So when we got these first 4 isolates, we looked at virulence and transmission in ferrets, but we only looked at contact transmission. And the reason why we just focused on that is up until 2022, the H5 viruses that had been characterized in ferrets largely showed very inefficient contact transmission from an infected animal to a naive animal, and really rarely, if ever, showed any signs of illness when the virus was transmitted into the contact animals. So we initially just set up to characterize this contact transmission. We infected ferrets, just 1 or 2 per cage. And this is done by giving them 10 to the 6 infectious units or plaque-forming units by the intranasal route. And then 24 hours later, we'll add 1 naive contact directly to the cage. We monitor weight and temperature and collect oral and nasal swabs every couple of days from all of the animals so that we can look at the patterns of shedding in the infected and contact animals. So it was quite interesting what we saw. All of these viruses are lethal in ferrets, and I think far more lethal than what we used to see with earlier viruses, H5 viruses. The only survivor we had was one of the animals that was infected with the blue-winged teal virus. But otherwise, lethality occurred in the infected animals somewhere between days 4 and about, I think, maybe 12 was the longest time to death. What really stood out here, though, is that this red-tail hawk virus and the red fox Ontario virus caused very rapid lethality in the infected ferrets. So basically by about day, I think 6 or 7 here, most of the animals are gone, and even as early as day 4 with the red-tail hawk virus. The other thing that was very striking, though, was that the red-tail hawk virus and the red fox Ontario virus were able to transmit into contact animals, and we saw disease with these animals. So with the red fox Ontario virus, there was at least 1 contact that became sick and showed signs of clinical illness, and this is characterized here by the weight loss that's associated with infection. With the red-tail hawk virus, it was even more striking because we saw lethality with this virus in the contact animals that occurred very quickly after they became infected. With the blue-winged teal virus and the red fox PEI virus, we don't see any clinical signs in the contact animals, but when we look at the pattern of shed viruses, we see that in the contact animals, so that's these 2 columns here, that all of the contacts were shedding virus at some point during the experiment, showing that they had become infected by direct contact. All of the infected animals, of course, also shed virus, and this is particularly striking with the red-tail hawk virus, where quite high titers were shed from all of the animals. So we wanted, because of this kind of unusual profile that we see with the red-tail hawk virus, to look at the possibility for aerosol transmission in these animals. And this is done very similar setup, except that we have this double wire mesh in the cage where the animals are co-housed that prevents the airborne contact animal from coming in direct contact with the infected ferrets or the direct contact. So in this experiment, an infected animal is co-housed with the direct contact on the same side of the cage. The airborne contact is on the other side of the cage, and it's downstream of the directional airflow inside of the cage. And then we just monitor the same way as we did in the previous experiment. And again, what we saw was very rapid lethality in the infected animals. 3 of the 4 infected or contact males, direct contact males, ferrets died, and 2 out of the 4 direct contact females died. But when we looked at shed virus patterns, we can see that with these animals that did get sick, that we see the contact animals were shedding virus, but none of the aerosol contacts shed virus. They didn't show any signs of disease. There was no weight loss or anything. So, it really looked like this virus is not being shed. And we use more sensitive assays than just live virus detection, since these values that I'm showing here are all detection of live virus using a quantitative PCR and did not observe any signs that the virus had transmitted. So there's really no airborne transmission. And we confirmed this by looking at seroconversion at day 21 when the study was ended, and there's no seroconversion as well. We set up a control experiment in this study as well with a pandemic H1N1 virus, just to be confident that we had everything set up in a way that we would actually see aerosol transmission if it could occur. So the pandemic H1N1 virus is from the 2009 pandemic, human virus that is known to transmit very efficiently in ferrets. And we only looked at the infection to aerosol contact animal transmission. And you see the infection in the infected animals, and all of the aerosol contact animals also shed virus. They all seroconverted, of course, as well. So we don't see any signs in this particular virus or with the viruses that we initially started looking at for aerosol transmission, but we still have an interest in knowing what are the molecular determinants that could really influence, enhance contact transmission. And we're going through the process now of getting approval to reconstruct viruses so that we can look at, the changing properties that are associated with different gene segments, since, it's a little bit of sensitive type of research, being that it could be construed as dual purpose or dual use research, right? So we're initially targeting things that might be more loss of function rather than gain of function, but. So this is something that we're just starting to work on with these viruses now. But I think one of the things that was really interesting that we kind of did as an extension of this experiment is that we had these 10 ferrets that were infected with the pandemic H1N1 virus. And they all survived infection, of course. So we wanted to ask the question, does prior immunity to pandemic H1N1 virus provide protection against these H5N1 viruses that are circulating now? And, the reason that we asked this question, of course, is because in 2009, it had been shown that the H1N1 immunity in animal models provided quite strong protection against H5N1 viruses that were circulating at that time. So we divided these 10 animals, so they were 5 females, 5 males, into roughly equal groups of male and female animals, and then challenged them with 2 different H5 viruses. One is an H5N1 virus, this red-tailed hawk virus that was very virulent. And then the other was an H5N5 virus from a skunk in Prince Edward Island. And this was another virus that had been transmitted into North America from the Eurasian source through the flyways that appeared in 2023 and caused a number of outbreaks in animals across Canada. And we had characterized it, shown that it was very virulent in ferrets. So when we challenged 5 ferrets with this H5N1 virus, they all survived infection. And we gave them a pretty high dose again. We gave them 10 to the 6 PFUs intranasally. We did see that some of the animals experienced some mild weight loss, and even by the end of the experiment at day 14, not all of them had recovered their original body weight, so they had experienced some level of disease. And the infected animals shed virus in both their oral and nasal swabs, so quite high titers of virus. So, this H1N1 immunity may not have prevented transmission of the virus, and wasn't completely successful at suppressing replication either. When we infected the animals with H5N5 virus, it was a little bit different. We saw that 2 of the female infected animals died. And they died quite quickly, almost as quickly as if we had used this virus to infect naive animals. And again, they shed virus. The other animals, the ones that survived, also lost some weight, and only one of them recovered significant body weight by the end of the experiment. But this does show that complete protection from lethal outcome was achieved against the H5N1 virus, and partial protection with H5N5. And this mirrors some studies that have been more recently published that show this kind of cross-protective response against or with H1N1 pre-existing immunity against H5N1 viruses. I don't think anyone has looked at H5N5 viruses yet. And it does also suggest that a matched N1NA may contribute to better protection against H5N1 than it does against H5N5, and that might be just because of this, degree of similarity between the N1 neuraminidase of the H1N1 and H5N1 viruses. So just coming back, this is my last slide on the transmission information. Did we ever see any signs of aerosol transmission in our animals? Because, if you've been following the literature, you'd know probably that there was some inefficient aerosol transmission demonstrated from viruses that were isolated in people that had become infected through their interaction with the infected cattle. And we actually did see a couple of viruses, both isolated from skunks. So skunk Alberta 22, which is an H5N1 virus, and skunk PEI 23, which is the same H5N5 virus that I was mentioning in the previous slide. We infected ferrets with these in the same contact plus aerosol transmission experiment that I described previously. And we do see virus that is shed from the infected animals, but the skunk Alberta virus is less pathogenic. So it only killed one of the contact animals. Both directly infected animals survived infection. So we only had this is kind of done at a low scale, so like just a simple pilot experiment where we have 1 cage of males, 1 cage of female animals, and one of the male contacts, direct contacts, died. None of the aerosol contacts showed any sign of disease, and there was no weight loss. In the case of the skunk H5N5 virus, all of the animals died by day 4 that were directly infected, and we didn't see any virus shed from either virus in the contact animals or the aerosol contact animals. But when we looked at seroconversion in all of these animals, what we found was that, with the skunk Alberta virus, both of the infected animals, both of the control animals, and one of the aerosol contacts had good levels of seroconversion, suggesting that the virus had been transmitted to the aerosol contact animal and the direct contacts, even though we didn't see any signs of replication in these animals. In the case of the H5N5 virus, we didn't have any data from the infected animals because they died too quickly, but both contact animals seroconverted and one of the aerosol contacts seroconverted. So this does suggest that there is efficient direct contact transmission and inefficient aerosol contact transmission with these viruses, which is similar to what was reported with the bovine viruses. It's occurring without changes in receptor binding, which would explain why it's so inefficient. But it's still of interest because any opportunities for a virus to transmit between mammals or kind of enhances the potential for it to acquire changes that could be a higher risk to humans. So just to summarize what I've been talking about, the clade 2.3.4b viruses exhibit higher virulence in animal models of infection. Overall, we've seen greater spread in poultry and wild bird populations, increased host range for both wild birds and mammals, and sustained replication in some mammalian populations. Reassortment and mammalian adaptive mutations contribute to the increased virulence. And of course, you can see this just based on the differential virulence and the effects that we have with the viruses, but there's really very little that's understood at this point about determinants that could be influencing that. So that is something that we're interested in looking at. And adaptations may help the virus overcome the barriers to transmission. It's not really clear how this might happen, but it could be due to infection of different cell types in the upper respiratory tract or just shedding much more virus than used to occur with some of the historical isolates. And it's occurring without changes in receptor binding, but this does enhance opportunities to evolve more efficient transmission and represents an increased risk to both human and animal populations. So for the second part of what I wanted to talk about, I was going to just discuss some of the vaccine work that we're doing. And this is just 3 projects that we've been working on with collaborators that have come to us with their vaccine platforms, just to give you an idea of like some of the work that we're helping others do that that can contribute to the response to an outbreak with these viruses. So the first one is a nanoparticle vaccine. So it's just a protein-based vaccine that was developed at the University of Buffalo in the lab of Jonathan Lovell. And for this project, we worked with Richard Webby at St. Jude Children's Research Hospital, as well as John Fulton here in Ontario at BioNiagara. So their approach is this kind of lipid nanoparticle that they have developed. And it contains a cobalt porphyrin complex that's embedded in the lipid bilayer. And what happens is that when protein antigens that have a His tag are mixed with these lipid nanoparticles, the His tag allows the protein to interact with the cobalt, and it anchors the proteins directly to the lipid nanoparticles. This happens spontaneously in solution, so you basically just have to mix everything together, and that's why they refer to it as the SNAP system or SNAP technology, spontaneous nanoliposome antigen particleization. And this is a very attractive platform for us because it's a really quick way to generate vaccines against emerging viruses, because one of the first things that we seem to see for product development is the production of viral antigens as proteins by a lot of companies that make proteins for diagnostic purposes and other projects. So this is something that I think could be used quite quickly against any virus that's emerging. So we use particles that they termed CPQ. So they have this cobalt porphyrin complex. And they also have in the lipid nanoparticles a couple of adjuvants. One is monophosphoryllipid A and QS21. So those enhance the immune response to the vaccine. And they used HA and NA antigens from clade 2.3.4b virus, Jiangsu 2023, that's very highly related to the red-tailed hawk virus that we had previously characterized. So we used the red-tailed hawk virus as our challenge in these experiments. We had 3 study groups, CPQ with just H5, or with just N1, or with H5 plus N1, and then a control group with just mice that were immunized with PBS. This is a fairly typical experimental setup for us for vaccines. In this case, we primed the animals with 1.8 micrograms of the vaccine. Our group sizes were 20 animals of equal males and females. We took blood before they were boosted, and then again before they were challenged. And they were boosted at day 21 with the same dose of vaccine, and then challenged with 10 times the lethal dose of this virus, which is just 10 plaque-forming units, because the LD50 is one infectious unit in mice. 4 days later, we take tissues from the mice. So mostly we're looking at titers in the lung just to assess the level of viral replication. And then the remainder of the mice are assessed until day 21 for survival and protection from clinical signs of disease. So looking at antibodies against H5 and against the N1, you can see that in the vaccines here with that have H5, they develop a good immune response that's strongly boosted by the booster vaccination. Of course, there's no H5 antibodies in the N1 group alone. And the similar is seen with the 2 vaccines that have the N1, although the overall titers are not as high as we see with H5. These antibodies against H5 are also neutralizing, so we do microneutralization assays with the live virus. All of the mice have strong neutralization titers. As I mentioned, N1 antibodies alone are not neutralizing, so we don't see any neutralization with the N1 vaccine alone in this particular experiment. So when we challenge the mice, Both of the vaccine groups that have the H5 vaccine in them are fully protected from lethal outcome and also fully protected from any weight loss. So there's no clinical signs of disease in these animals. With the N1 only vaccine, there was 70% protection, which is actually pretty good for an N1 vaccine. And when we look at lung titers at day 4 in these vaccine groups, the H5 groups, so the H5 or the H5 plus N1 groups, we couldn't detect any live virus in a live virus assay, at least up to the limit of detection. And with the N1, we also saw pretty good protection in most of the groups, although 2 of the animals, there was breakthrough, so there was fairly high titers of replication. And this is probably what's also happening with these animals that succumb to infection after challenge. So we are expanding our interest in this for an H5 vaccine. So right now we're using just the H5 plus N1 vaccine. We're testing various dose ranges. We're also testing a single dose to see whether we could achieve protection after like 14 to 21 days. And this is an important consideration, of course, for a vaccine that could be used in an emergency situation. So, and as I mentioned now, we think it's also an adaptable platform for emerging viruses. So we have an interest in looking at this same platform for filoviruses and Nipah virus and other viruses that we're working with. So another project that we're working on is a mRNA vaccine that was developed at the University of Cambridge by Jonathan Heaney's group. And so they have a little spin-off company called Diosynvax, and they specialize in the development of computationally optimized vaccine antigens. So they're basically making vaccines where they study important epitopes for protection and doing things like deglycosylating the antigens, just ways that they can strategize to improve the antibody responses to their vaccine. And their objective is to provide a single vaccine option that is protective against all H5 viruses. So in Canada, they're doing this as a collaborative effort with some Canadian companies and the University of British Columbia. And they have this little collaboration called the Canadian Pan-H5NX Vaccine Manufacturing Consortium for Pandemic Preparedness. So Jonathan is Canadian, so he has an interest in just working with Canada and kind of amplifying the science that's being done in Canada as well. So, so they also work with a number of other viral platforms, and they're just testing out different platforms with other viruses. In this case, it's an mRNA, but they also have used DNA vaccines and poxviral vectored vaccines and things like that. So the reason that they're looking at ways to expand the breadth of protection is, of course, because as H5 viruses circulate in different animal populations, they evolve, and they evolve particularly immunologically because there's a lot of pressure in infected animals on the antibody binding epitopes. It's also the case that there's a lot of other H5N1 viruses that are in circulation or that have circulated in the past. And I've only been talking about the 2.3.4b viruses right now, but another example of an important pathogen is this clade 2.3.1c virus that's been circulating in Southeast Asia. And over the last quite a few years, it's been causing quite a few cases with lethality in humans. So this is another potential candidate for an H5 virus that could emerge as a novel pandemic strain. And there's a lot of other clade 2 viruses in circulation. Clade 1 viruses, for the most part, I think have probably been replaced by the clade 2 viruses. They're probably not circulating anymore. But we did use this as one of the viruses in the study as an example of a relatively highly divergent virus in the H.B.A. So as I said, the objective here is to plan for a single off-the-shelf vaccine that could be used against any emergent H5 virus without knowing in advance, of course, which virus that could be. So to do this study, we wanted to validate cross-clade protection of the Diosynvax H5N1 vaccine in mice. And we set up three identical vaccine groups and then challenged them with three different challenge viruses that represent viruses of different diversity in the immunological epitopes of HA. The particular virus that they formulated expressed their computationally generated H5HA. They used an N6NA because in the past, H5N6 viruses have also been prominent in humans as a source of viruses, or as a virus that can infect humans and cause disease. And then they included the M2 antigen, and that was the one I mentioned previously that's very highly conserved between all the viruses. And they're using a novel LNP that was developed by the Canadian company that's just believed to exhibit low toxicology compared to like vaccines that were used during the COVID pandemic that were I think, they had things like PEG in them and other things that would cause like more toxic responses to vaccination. We compared the vaccination to a candidate whole inactivated virus vaccine. So whole inactivated virus vaccines are, of course, the vaccine that we all get as a seasonal vaccine, and they're the vaccines that are typically stockpiled by other countries for pandemic response. And in Europe, I think the vaccine that's probably stockpiled by most countries is this one called Astrakhan 2020 against an H5N8 virus from Russia. And this virus is, or this vaccine is commercially available, so it was of interest for us. It's a clade 2.3.4b virus, and we used three different challenge viruses, a clade 2.3.4b virus. And this was actually the patient virus of, I don't know if you, the teenager that was sick in British Columbia that eventually recovered from her infection. But this virus is very pathogenic in mice. We used a clade 1 virus, which was isolated from Vietnam in 2004, so this is another human patient virus. And then we used a clade 2.3.21c virus from Cambodia, another human patient virus from a case, a person that had died. This experimental design is very similar to what I showed you before, basically identical in all the timing of the events. And when we did this study, we found that the Diosynvax pan H5N1 vaccine protected very well against all the challenges. All of the animals were protected from lethal outcome, and all of the animals were protected from any signs of clinical disease. So there was no weight loss, there's none of the neurological signs that can be associated with infection in the brain and things like that. So we also looked at virus titers with these animals, and they had basically no virus titers that were detected in any of the tissues. And this was true against all of the challenge viruses. If we compare this to the whole inactivated virus vaccine, this vaccine provides very good protection against the clade-matched 2.3.4b challenge. But as the challenge virus becomes more antigenically diverse, then we start to see a drop-off in the level of protection. So with the clade, the Cambodia virus, the 2.3.21c virus, there was 70% protection, and we see some clinical disease in these animals. And with the Vietnam virus, the clade 1 virus, protection was at 50%, and there's quite a bit more disease observed in these animals as well, shown here by the weight loss. So this shows that their H5 or computationally optimized vaccine is expanding the breadth of protection against against H5N1 viruses in general. And so one of the plans to do next is to test this vaccine in ferrets, because ferrets are much more difficult to protect than mice. And they would like to proceed to clinical trials with this vaccine if the ferret data works out really nicely. So, and then the last vaccine that I'm going to mention is a VSV as an example of a attenuated viral vector vaccine. So VSV is vesicular stomatitis virus, and it's the backbone of the Ebola virus vaccine that was developed at the NML that has been approved for use in humans in outbreaks of the Ebola virus. And this platform actually builds on that Ebola virus vaccine. So it uses the Ebola virus glycoprotein in the place of the VSV-G protein. So this attenuates the virus, but it also supports viral replication because Ebola virus G can support viral replication. What's also interesting about the G protein, Ebola G protein, is that it has this region called the mucin-like domain that can be either fully removed or can be replaced by other amino acid sequences, and it's still biologically functional. So it's, in this case, in this vaccine, was used to put in a tetrameric M2E antigen. That's just the tetrameric extracellular domain of M2E. The other thing that's very important about the Ebola virus GP is that it targets Ebola virus to infection in macrophages and dendritic cells. And this improves antigen presentation because these cells can operate as antigen presenting cells. And, so then it's targeting the VSV vaccine to these cell types. So that boosts the immune response that's seen with the vaccine as well. And finally, the H5HA of the red-tailed hawk virus was added as an additional cassette. And this work was done by Dr. Yaojian Yao at the University of Manitoba, and something that he's been working on for quite a few different viral pathogens. So again, we had the same vaccine design as I've showed you before, but the difference here is that we had two different vaccine routes. We did either an intranasal vaccination group where they were given 10 to the 5 infectious dose or units of this vaccine, or an intramuscular route where they were given 10 to the 7. And we picked these doses based on work that they had done previously. So they knew that these doses should be protective in their models against influenza infection. Again, we see good IgG titers against HA in both the intranasal and IM vaccinated groups, but the boost effect at day 21 is much more is much stronger with the IM vaccine, and that could be due to the higher dose, or it could be the difference in the route of vaccination. But when we challenged mice with these with the red-tailed hawk virus, they were all equally protected in both vaccine groups from lethal outcome. There was no signs of clinical disease, and we looked at titers of virus in the lung and a few of the peripheral tissues that the virus will spread to, and there was no signs of any level of replicating virus in either vaccine group. So again, this virus, this vectored virus vaccine is very potent, and so we would like to expand now the analysis to other clades, like the more divergent clades of H5N1 virus, and potentially also look in other animal models to see how good protection is. So just to wrap up the experimental vaccine, there was 3 of them: the VLP nanoparticle, that's highly immunogenic protein-based vaccine, and we like it for its adaptability to emerging viruses. The Diosynvax Pan-H5NX mRNA vaccine as a potential off-the-shelf mRNA vaccine candidate. And then VSV, which is a fairly potent vaccine candidate with enhanced antigen presenting cell targeting. And the M2E that's present in this may expand the immunogenicity to more divergent viruses. So that is something, like I say, we'll be interested in looking at. So I'd like to just end there with a slide to acknowledge some of the other people at the NML in the special pathogens group, the influenza respiratory viruses, coronaviruses group at the NML, and of course Johannes's group at NCFAD. And so thank you for your attention, and I'd be happy to answer any questions.